Good morning, everybody. Let's go ahead and talk about the week we have ahead of us and then jump into our lesson on complex inheritance and human heredity. So, Monday, there is a crash course genetics ed puzzle video that you should get on and watch and answer the questions. Tuesday/Wednesday, you should get on and make sure you complete the chapter 11 vocabulary assignment. Just define each word, use it in a sentence, do a Google image search to give an image of it. The same old thing that we usually do with new vocabulary words. And then Thursday, there are some human inheritance questions. And again, these questions don't necessarily have a right or wrong answer. I mean, it does have what I'm looking for, but mostly you're going to give me what you think is the answer. What is your best hypothesis on these questions? What do you think the outcome is going to be? And then there's no school Friday this week, as well as no school Monday the next week. So you get a nice 4-day weekend. That's basically it for this week. So let's go ahead and jump into our lesson on complex inheritance. So we've been talking about all of the different things that go into basically what makes an organism exactly specifically have the traits that it has. We've talked about what alleles are, different forms of the same trait. We've talked about what it means for something to be dominant or recessive. We've talked about the difference between homozygous dominant things, heterozygous dominant things, and then recessive things. Well, today we're going to look at what are some of the different things that could be going on within our genes. These things are a little bit more complex. Last time it was pretty basic. This is a little more complex, but let's go ahead and jump right in here. So we have these things called recessive genetic disorders. Some things that happen in our genes are dominant, and the dominant things are kind of the more preferred things. But every now and again, two recessive traits combine, making that homozygous recessive trait. And when these recessive things are expressed, sometimes that's not good. So a genetic disorder that is coming from recessive genes is expressed when an individual is homozygous for those recessive traits. Those with at least one dominant allele, they're not going to express this recessive disorder. But every now and again, if they become heterozygous for this recessive disorder, they are going to express it. Or if they become homozygous for the recessive disorder, they're going to express it. And if they're heterozygous for it, they still carry it and they can still pass it on. They're just not personally going to express it. One of these disorders is cystic fibrosis. So this disorder affects the mucus-producing glands, digestive enzymes, sweat glands. And chloride ions are not properly transported out of the cells of a person with cystic fibrosis, and it causes mucus excretion that clogs ducts in the pancreas. It can interrupt digestion. It can block respiratory pathways in the lungs. It is not good. It is not fun. A person could also have albinism. This is caused by altered genes that result in the absence of skin pigment, melanin in hair and eyes. Individuals with albinism have very pale skin, white hair, pink irises. Again, this comes from recessive traits being expressed. We have Tay-Sachs disease. This is caused by the absence of the enzymes that are responsible for breaking down fatty acids called gangliosides. And gangliosides accumulate in the brain, inflating brain nerve cells and causing mental deterioration. Again, this is not a good time. This is not good. We have galactosemia. This is a recessive genetic disorder characterized by the inability of the body to digest galactose, so that you can't have milk products. Now, some dominant genetic disorders, this means it doesn't require a recessive trait to be expressed. This is just going to happen if you get those genes and they are dominant. We have Huntington's disease. This is going to affect the nervous system, causing gradual loss of brain function. It occurs in about 1 out of every 10,000 people in the United States. We have achondroplasia. It causes small body size and small limbs that are comparatively short. It's caused by an abnormal gene that affects bone growth. Okay, well, one of the things that we can do to trace inheritance of traits through several generations is come up with what's called a pedigree. And a pedigree is a diagram that traces basically any trait and how it could be inherited through several generations. Typically, a female is a circle, a male is a square. They shade it in dark if the individual expresses the trait that's being studied. They shade it in half and half if it's a person that's a carrier, but they're not expressing the trait. Straight lines left to right indicate that these are the parents of offspring. Lines that go up and down means you're jumping to the next generation. And then if you have these, several arched connected lines, 1, 2, 3, 4, that's going to indicate that these are siblings being talked about. Now, numbers are just each individual, 1, 2, 3, 4. Roman numerals is each different generation. So like 1-1 would be the dad in this case, 1-2 would be the mom, 2-1 would be a female child, 2-2 would be a male child, and so on and so on. That's, if you know how to read these symbols, you can read a pedigree. Now, pedigrees can be used to examine both recessive and dominant genetic disorders. Information about an individual's genotype can be inferred from the phenotype of the parents and the offspring. So you can make a good guess is what that means. You can look at the parents and make a pretty good guess what the offspring are going to have. Now, inferring genotypes, what we mean is knowing physical traits can determine what genes an individual is most likely to have. You can't 100% predict it, but you can make a pretty good guess. Record keeping helps scientists use pedigree analysis to study inheritance patterns, determine phenotypes, ascertain genotypes. This is why if you've ever had to go to the doctor and fill out those forms, they want to know your family history, who in your family has ever had cancer, heart problems, lung problems, all these things. They want to know that stuff because it gives them a pretty good guess, not an accurate 100% correct prediction, but a pretty good guess as to what things might be going on with you or might in the future go on with you. Now we have something called incomplete dominance. Some organisms, sometimes in some organisms, heterozygous individuals will display the dominant phenotype. Now with incomplete dominance, the heterozygous phenotype is an intermediate between two homozygous phenotypes. And this example here shows us red is crossed with white. You would think, okay, the offspring is going to be either red or either white. Well, when the dominance is kind of incomplete, the plant is pink. It doesn't completely show either traits. It shows parts of both traits. And we all know red and white make pink. So that's a good example of incomplete dominance. It doesn't completely show one trait over the other. It incompletely shows both traits. Codominance is when both the dominant and recessive alleles are expressed in a heterozygous individual. If you can think of, have you ever seen a puppy or even a person with different colored eyes? That would be codominance. The dominant and the recessive allele was expressed in this individual. Sickle cell disease comes from a codominant issue. Changes in hemoglobin cause red blood cells to become sickle-shaped. People who are heterozygous for the trait have both normal and sickle-shaped shells. Sickle cell disease and malaria. So those who are heterozygous for the sickle cell trait have a high resistance to malaria. So there is a positive thing coming out of that if they were to ever be exposed to malaria. The death rate due to malaria is lower in areas where the sickle cell trait is higher, meaning more people live to pass this on to their offspring. Now, sometimes we have multiple alleles. So some forms of inheritance are determined by more than two alleles. If it takes more than two alleles to determine something, we call that multiple alleles. This shows up in like blood groups and blood types in humans. The ABO blood group has 3 forms of alleles. Sometimes we call those AB markers. In rabbits, the color of their fur comes from multiple alleles. If you can picture a pet, picture a rabbit, you've probably seen a solid white rabbit, a solid black rabbit, gray, brown, whatever. But sometimes they have splotchy fur with multiple colors in it. And you can tell the hierarchy of dominance based on the colors. Like in rabbits, 4 different alleles code for coat color. And the hierarchy goes, C over CCH over CH over C. And the presence of multiple alleles increases the possible number of genotypes and phenotypes. So whichever one of these they have the most of, they're going to have the most of that color, but they could still have a combination. So you could still see many colors at the same time. That's why pets, unlike humans, pets, certain animals can have many colors in their fur. Okay, we have a scenario, a situation, whatever you want to call it, called epistasis. This is an interaction where one allele hides the effects of another allele. One good example is the fur coat of Labrador retrievers. We have a dominant allele, let's call it E. It determines whether the coat will have a dark pigment or not. We have allele capital B, determines how dark it'll be. And when a dog has recessive little e alleles, the coat will be yellow because the little e allele masks the effect of the capital B allele. All that to say is sometimes alleles can hide effects of other alleles if they overpower them. And then we have what's called sex determination. One pair of chromosomes, or the sex chromosomes, determine an individual's biological gender. In females, it's XX. In males, it's XY. And the other 22 pairs of chromosomes, we call those autosomes. So each human has 23 pairs. 22 of those pairs are autosomes. One of those pairs is your sex chromosomes. The X chromosome carries a variety of genes that are necessary for the development of females and males. And the Y chromosome mainly has genes that are relevant to the development of only males. So in females, one X chromosome is inactivated in every cell. We call this dosage compensation or X inactivation. Whichever one of the X's that's going to stop working is random. It doesn't really matter. It just randomly happens. So here's kind of a diagram. We have X chromosomes. One of them's active, one of them's inactive. So in females, it doesn't really matter. We have these things called bar bodies. The inactivated X chromosome is observed, and under a microscope, if you stain the slide darkly, you can see the inactivated X chromosomes. And one of the names we call that inactivated X chromosome is called a bar body. So if you were to observe under a microscope the inactivated X chromosome, we call those bar bodies. Now, some traits are controlled by genes located on the X chromosome, and those are sex-linked traits. Because males have only one copy of the X chromosome, they are more affected by recessive X-linked traits. So males cause this to happen because they have only one X chromosome. Things like color blindness, red-green color blindness, it's on a recessive X-linked trait. Mothers are the carriers in this case. Hemophilia, it's a recessive thing. It's X-linked trait that causes delaying of clotting of blood. So you do not want to get cut or wounded because you're going to bleed out. Now, some traits are polygenic, and this arises from the interaction of many multiple pairs of genes. Things like skin color, height, eye color, these are polygenic. They're not simply one allele. It's actually many alleles. If you look at the shades of skin color, we all know there's so many different shades of skin color. It's because the combination is vast. There can be many different combinations, which is why there are many different skin colors. Same with eye color, different heights, all of those things. Now, all of those are automatically from your genetics, but we also have things from the environment that can influence genetics. Sunlight and water are one of these examples. Without enough sunlight, plants are not going to produce flowers. Without enough water, plants are going to start losing their leaves. Most organisms experience a phenotypic change if there's extreme heat. Now, some of these weird genetic occurrences have been able to be studied in twins. So these twin siblings get studied to see what would happen if one thing happens to one and one thing happens to the other. These twin studies have helped scientists separate genetic contributions from the environment versus what's strictly genetic. Traits that appear frequently in individual twins are least partially controlled by heredity, and traits expressed differently in the twins are strongly influenced by the environment. So they're able to tell, okay, this is based on your genetics, or this is based on factors we influenced you with. Then we have karyotype studies. Scientists study not only genes, but entire chromosomes. And images of chromosomes stained during metaphase can allow scientists to study sister chromatids and homologous chromosomes. A karyotype is a micrograph, or this graph here, this image, in which the pairs of homologous chromosomes are arranged in decreasing size. So they match them up, the equal ones, and then arrange them from biggest to smallest. In our chromosomes, we have these things called telomeres, which are the protective caps on the end of chromosomes. These telomeres consist of DNA and proteins, and they serve as a protective function for the structure of chromosomes. They help them maintain their structure. Okay, we have another situation called nondisjunction. This happens during cell division where sister chromatids fail to separate properly. When these sister chromatids don't separate, you can look down here, you know they're supposed to separate into equal parts. Well, here's a situation where one of them, it just doesn't. When it passes on these gametes to the offspring, it's only supposed to pass on one individual sister chromatid, but it passes on multiple. So when this joins up with the other individual's gamete to start producing an offspring, there are going to be too many chromatids, too many chromosomes. So one situation that you can probably think of would be Down syndrome. In Down syndrome individuals, there is an extra chromosome on the 23rd chromosome. This is all stemming from what's called nondisjunction. Those chromatids just don't separate properly in the formation of the gametes, and then they get passed on, and the new organism has too many chromosomes. So again, Down syndrome comes from an extra 21st chromosome. Characteristics of a Down syndrome individual are usually distinctive facial features, short stature, heart defects, sometimes a mental disability, but not all the time. But these are, again, the result of that non-disjunction happening, and then too many chromosomes get passed on. Fetal testing is a thing that can happen for parents. Fetal tests can provide information on potential genetic disorders and chromosomal status of developing babies. A doctor can just take and inject a needle into the mother and take a sample of the blood and the amniotic fluid and all these things and figure out what disorders may occur. Okay, that was a lot of facts about complex inheritance and complex human heredity. If you need to go back and review some of that, feel free to rewind this video, watch back through some of the parts and listen again. Otherwise, good luck on your assignments for this week. And as always, please feel free to ask me for help. Send me an email, raise your hand if you're in person. But let me know if you need anything at all, and good luck.